I am coming from the area of disparity science, social science, and Alzheimer's research. I have met many of you before, mostly online, but I've certainly learned a lot from listening to the conversations today, and I'm pleased to say that we are making some very innovative headways in thinking about disparities, thinking about linkage of exposome to action, as well as thinking about life course. So this is the beautiful University of Wisconsin. We have a number of other people here from the Big Ten, so very happy to be here. So in my outline today, I've been asked to talk about the neighborhood study, which is an NIH-funded study, and we will go into some depth on that. But in addition, I'm going to talk about the importance of data democratization in order to link to real-world change, and give you many examples of how in the United States, social exposome data is being used in policy and community constructs in order to target resources more effectively. So the core construct of all we do at the Center for Health Disparities Research lies on this question. Within societies, we do an excellent job of improving health if you live in a place that looks like this. You can have access to high quality medical care, be able to live longer and live healthier lives. But if you live in a place that looks like this, the story is very different. You will live shorter lives, you will have more diseases, and you will ultimately have more disability. And of course, in the field of Alzheimer's disease, I could show you hundreds of US studies that show this linkage, this disparity for individuals who live in places like in that second slide, and increased risk of cognitive disorders and brain diseases. But I'm going to show you two excellent studies from outside the United States. The first comes from the English Longitudinal Study on Aging, which suggests almost 70% increased risk of dementia incidence in those with the lowest wealth, independent of education and other factors. And also the wonderful Chinese Longitudinal Health Longevity Study, which shows that poverty is associated with a 34% increase in incident cognitive impairment. This issue of dementia diagnosis and cognitive impairment linking to these social factors has been echoed. It is definitively proven at this point. When we think about why health disparities occur, often in my field, we think about these types of frameworks. These are the National Institutes on Aging, National Institutes Minority Health and Health Disparities also has a framework that explains health disparities, and so much of this is a focus on the social exposome. So when they are thinking about health disparities, or my field is thinking about health disparities, we think about different environmental, sociocultural, behavioral, and biological factors that interact with each other across an entire life course to either produce health or disease. And when we think about those social factors, that's that leftmost column there, you can see many factors other than the ones that were talked about today. Each of these bullet points within these bubbles is supported by entire bodies of literature. So this is a huge field. There are strong evidence to support the linkage of these different factors with health. And in particular, we are going to focus on socioeconomic factors in the rest of my talk today. The social determinants of health are those factors that include income, education, housing quality, and employment, and ultimately promote either health or disease. The social exposome can be quantified, it can be studied, and it can be modified. Pulling this beautiful picture from Dr. Jett's work, of course there's many others that I could show here as well. But ultimately, measures of the social exposome exist because the fields of demography, sociology, and social sciences have been around for 100 years. We have highly reliable measures of these constructs that can ultimately link to biological outcomes. But often they've been forged within silos that do not necessarily communicate all that well to the other areas. The other thing that's very important to realize is that individual level social factors, the factors about me or about you, the things that are our own social traits, are independently related to health from exposome level social factors. So the neighborhoods that we live in, the environments that we live in, those types of social exposome experiences impact health independently of the individual level factors. There have been many, many natural randomized studies to support this, and hundreds of work in the recent about 5 years showing this independence within brain health in particular. It doesn't mean that one is more important than the other, but we don't just map things to take the place of an individual level survey. They both are important and they both predict health in different ways. So our team has, for the last about 20 years, been working on a metric called the Area Deprivation Index. This was a metric that was originally developed by HRSA, the Health Resources and Services Administration, back in the 1980s. But its development was not quite ready for linkage to biological outcomes because of the geographic size that it was using and the old forms of data. So we received a grant from the National Institutes of Minority Health and Health Disparities to update this metric to a much more geoprecise level, to bring it to more modern data sources within the United States, and to validate it across a whole host of poverty-linked health outcomes. It is an incredibly widely used measure at this point. Our team at the University of Wisconsin produces this measure each year. It includes 17 different measures of the social determinants of health, including income, education, housing quality, and employment, that are built at the census block group level, which is about 1,500 persons. The methodology has been harmonized to international multi-domain indices of deprivation, which are used in the field of economics, often used in government to target resources. And so with this, we have done multinational studies with our colleagues from a number of different areas, particularly in the brain health space. It's a ranked score, it's time concordant, and it's strongly linked to health outcomes. Just in the last 2 years, over 1,500 papers have used our metric to be able to link social factors to biology and also to disease. We also know that the dosage and timing of these exposures changes across a lifespan and are measurable. And I'm going to show you some data on that in just a moment. And Bob's comments about this importance of lifespan and being able to track where someone has lived across a life course is absolutely critical, and our team is doing that now. The ADI has been recognized by US professional research and governmental bodies. It's been endorsed by the Department of Health Services Office of Assistant Secretary for Planning and Evaluation as one of 2 best measures for resource targeting across the United States. It's been endorsed by the Society of Thoracic Surgeons and many, many, many other groups. I will share more about the practical applications of this metric in the second half of my talk. But for now, we're going to turn and think a little bit more about the science. So when we think about the high ADI neighborhoods across the United States, they tend to be in the inner cities, they tend to be Native American communities, and they tend to be in highly rural areas. In this area of social factors and social exposome, previously the county level had been the gold standard, and many of you may know that the University of Wisconsin is also the home to a tool called County Health Rankings, which is a very helpful tool funded by the Robert Wood Johnson Foundation and used across a whole host of public health activities. But when we look at the county health rankings and we think about this need for greater precision when we're linking to fundamental biology, you can see some differences here. So for instance, Milwaukee County, where the arrow's pointing at there in southeastern Wisconsin, is a geographically smaller county, but it contains some of the most challenged areas within the state of Wisconsin. It has the highest rates of racial and ethnic disparities. And it ranks as highly disadvantaged on a county ranking. However, when we use a much more geoprecise measure at the census block group level, this ADI, you can see that the pattern across the county here, which is shown in a larger format here on the other side of the slide, shows variability across that entire region. The red areas here are the areas that are highest in ADI, the most disadvantaged. The blue areas are the least. And within a single county, we not only have the richest neighborhoods within the state of Wisconsin, but we have the poorest as well. So that geoprecision is absolutely fundamental for these social factors. And assuming that a 5-digit zip code or a county can replicate the true experience of a social exposome is a mistake that we like to ensure doesn't happen. In our own analyses, when we move the construction of the ADI up into higher geospatial units, its precision and ability to predict different types of, for instance, neuropathology and different types of health outcomes diminishes. The other interesting thing about these red areas is they are the historically redlined areas of Milwaukee, so this does link very tightly to past policy. The ADI, or this construct of social exposome, is now embedded within many brain health studies within the United States. It is now part of the Alzheimer's Clinical Trials Consortium. When patients are enrolled into those clinical trials, their address is pulled in and the ADI score is automatically pulled in as well. We have also worked closely with the teams at the Adolescent Brain Cognitive Development Study, in which they have shown strong association with living in a high ADI context and changes in the way the neuroconnectivity develops within the brain as young as age 2. We also work very closely with different pharma groups that are interested in clinical trials targeting as well as secondary analysis of these data on the basis of ADI. As I mentioned, there's over 1,500 papers just in the last 2 years, obviously many more than I can list. But to date, living in these highly disadvantaged areas by ADI, by social exposome, is linked to increased dementia diagnoses within the Veterans Affairs system. It's linked to cognitive decline. I'll show you some data on the AD biomarkers in a moment. It also has now been linked in multiple different studies to epigenetic age acceleration, increased rehospitalization, greater costs of care, poor care quality and access out of some studies out of UCSF. Out of a study, the SAGES study out of with Sharon Inouye's leadership out of Harvard, showing that individuals who are coming into elective surgery who live in one of these highly disadvantaged contexts by ADI have markedly increased risk of poor postoperative outcomes. On the basis of this, they are targeting patients now to try to increase the amount of social work support, nutritional support, and other activities in order to improve, ultimately, these post-surgical delirium risks. Increased cardiovascular risk, heart cancer, diabetes, and obesity, some more work out of Harvard. And out of a beautiful study done out of Yale, led by Tom Gill, showing that a cohort that has been followed for many, many years, those individuals that were living in the red areas, these high ADI contexts, not only lived shorter lives, but they were more disabled at the end of their lives. They needed more in-home supports and services. In our own work, we have been interested in linking these types of social factors to fundamental neurobiology. That has been incredibly difficult because the infrastructure does not typically exist. When we are talking about social factors, they tend to be created and used by social scientists. And my bioscience colleagues in the field of neurobiology in particular often do not have an awareness of these factors, nor do they necessarily have infrastructure in order to make those linkages. So we are moving in that direction aggressively now. This is some work that was published in JAMA Neurology in 2020 out of my team, looking at individuals who are cognitively normal. This is a cohort of individuals followed within the Midwest. And what we were able to show after full adjustment for all the things that we adjust for in the Alzheimer's field is that those individuals living in the highest ADI context not only had smaller brain volumes, but they had smaller hippocampal volumes, which is the seat of memory. The brain volume differences could be explained on mediation analysis by cardiovascular risk, which was fascinating, but the hippocampal volumes could not. Thinking that there may be different mechanisms in place, potentially reaching into childhood and early life. So that last study was cross-sectional. We were also interested in looking at longitudinal effects. So using the same study, following these individuals for 10 years, we compared the neuroimaging of these individuals at the point of entry to the study to where they were 10 years hence, and looked at where they lived across that lifetime. What we found is individuals who were living in the most disadvantaged context by ADI showed faster cognitive decline and greater loss in brain volume. Their brains were shrinking more quickly than their counterparts who lived in the wealthier context. So something, of course this is an association, but something about these contexts was leading to more aggressive cortical thinning. But also within my field, a large proponent of the work that is done in the Alzheimer's space is with brain tissue. In fact, we have entire diseases that we cannot identify during lifetime yet. We do not have in vivo biomarkers to be able to say, you have this disease or you have that disease. We have to look at brains that are donated at the point of death and then look at that tissue under a microscope to understand exactly why the patient was having the deterioration that they had. So linking biorepositories to social exposome data we saw as the next frontier in order to be able to help us understand more about why these associations might be occurring. Our first study on this, published in JAMA Open, was simply a feasibility study in which we partnered with the University of California, San Diego Brain Bank, one of the largest brain banks within the United States, as well as our own at the University of Wisconsin, to see if we could collect address data at the point of death and to then do a time concordant linkage to ADI using geocoding methods. With 453 brain donors in this pilot study, they had donated their brains from 1993 to 2016. There was no social factor characterization whatsoever on these tissues. It was merely very deep biological phenotyping. And what we were able to do was, first of all, find that this was a feasible approach. We were able to get their residential address from papers that were collected at the time of their donation. We were able to link it to a time concordant ADI. We can build ADIs back to the early 1900s. And what we were able to show is that for each step up that an individual lives, each step, each decile higher in ADI, each more challenged step up in more neighborhood disadvantage, the odds of AD neuropathology, the burden of AD neuropathology also increased in a dose-dependent manner. On the basis of this, we wanted to next move into the life course space. And using the studies that I just shared with you, and with this intent to move into the life course space, we were funded on the neighborhood study by the National Institutes on Aging. This study includes 23 different sites across the United States, and it is designed to examine the impact, mediators, and moderators of life course exposome on ADRD pathologic features, vascular burden, and cognitive decline. We are just in year 3 of this study, and already it's producing very interesting findings. These are our sites. This will include over 10,000 brain donors, so it will allow us to do deep characterization, as well as nearly 7,000 living participants. And for each of these, we will be doing deep residential history. So in order to allow this to come together, the first 2 years of this study were really about building the infrastructure to allow for protected health information data to flow into the University of Wisconsin. So I worked very, very closely with more lawyers than I can count at these 23 different sites in order to develop data use procedures and a cloud-based cybersecurity platform that allows all of these data to come together. The goal is for us to be able to process the data in this very secure environment, to build the residential histories, but then to de-identify that data and process it out for linkage to our other AD resources within the NIA, particularly core resources and others. Many of these resources are focused on drug development, they're focused on genome, they're focused on different biorepositories, and none of them have interest in geocoding or PHI data. So to be able to do that will increase the ability of our social exposome colleagues, or all exposome colleagues, to think about this area. So as I mentioned, we are doing these life course residential histories. We are just launching the living cohort now. That is very straightforward. We use standard survey techniques. It is a very big data cleaning exercise, and we do the geocoding back to the point of birth. But for individuals who have already passed and donated their brains, we do public data tracing. So this is from a paper that was published just this past year in Alzheimer's and Dementia, which shows our test cohort. The youngest was born in 1895. They were all dead. The earliest death was in the 1990s, all the way up to 2018. I hired a team, I have about 30 people on this team of archivists who go through and find the information about these brain donors within public record. They search city directories, census, military records, pension records, marriage and death certificates. We pull all of this together in a beautiful archive. Each of these documents has an address. We can create the residential history from these archival sources. and ultimately form the framework for these decisions in that way. In addition, all of these records contain other information, as you might imagine, including occupational history and other types of really interesting family and social connections that are going to be available for research as well. In terms of public data tracing, we have compared this to survey in a consenting living cohort group, and we're able to find that 73% of all person years are traceable, are foundable within, are discoverable within the public data. And this is almost 100% concordance with survey data. In fact, we can see where a survey participant is having some recall bias earlier in their life. We think the public data is actually better. But the process on this, the way we have done this with dual archival tracing, has been published by one of my MD PhD students in Alzheimer's and dementia just a few weeks ago. So when we look at this life course, you may say, well, what does this look like? And I'm purposely putting this slide up here, and I know it's a little messy, but it gives you a sense of the kinds of life courses we can produce. So subject age is on the horizontal axis, and ADI, this metric that I've been talking about, is on the upright axis. And the first thing to note is that individuals don't tend to always stay in the same strata of ADI, at least those within this cohort. There are a number of individuals who live their early life within high ADI context, and ultimately at the point of death, when they donated their brain, they had moved into a lower ADI context. So they started out perhaps in a poorer area, moved into a more wealthy area. So complicated life courses, but when we look at these life courses across an entire lifetime and link them to the findings in these brains, what we can say is that the prediction ability is interesting. So this was some work by a PhD student, Sarah Keller, looking at VBI, vascular brain injury, which includes brain tissue infarcts, microinfarcts, hemorrhages, and white matter rarefication. And what she was interested in is looking at childhood versus midlife versus late life. And first of all, individuals who either stayed in a high ADI context across their entire lifetime, that's the stable high group, or those that were increasing. So they started out in a low ADI context, moved to a high ADI context, had marked prediction of having higher rates of these VBI markers on their donated brains. And this was from childhood to midlife. When we look at childhood to older adulthood, again, thinking about this entire context now, if someone was living in a stably high ADI context, it may not be the same neighborhood, but their score was high across their life course, they had an odds that would approach 10 in terms of risk for these brain tissue infarcts. So the power of this technique we're just learning more about now, the potential to think about these life courses from advanced statistical methods is at hand. Additionally, as I mentioned, when we go through these archival documents, we find all sorts of other things, not just where someone has lived. And this is an assistant professor, a junior faculty who works on my center. His name's Dr. Ryan Powell, and he's interested in military history. And certainly when looking at some of these pilot data, those individuals, we had a lot of individuals from UCSD. UCSD is a big Navy site, for those of you who may not know. 60% of the donors had some level of military service on the basis of their public archival tracing. And this was directly associated with a markedly increased burden of both amyloid and tau in the donated brains. The next step here is to link to DoD records to understand more about different exposures and different areas of service within the military. So he's developing a grant on that now. But here's the real question, bringing this back again. How is this all going to improve health here? Coming back to the discussion at the end of the last talk, this becomes very important for us at a Center for Health Disparities Research. And it really involves us linking these types of research findings directly to action. The social exposome gives us a path for that as well. But to do so, we have to embrace a construct called data democratization. Data democratization is the step beyond open science. Open science is about sharing data for other researchers. Data democratization is about making data available for everyone within a society, regardless of their background, amateur or professional. We are interested in reaching individuals that would never set foot in an academic sphere. We are interested in reaching individuals that are actually crafting the policy and delivering the interventions in communities. And to do this, we built a tool called the Neighborhood Atlas. This is a tool that is supposed to be incredibly simple. And it is simple. You look at it and you think, oh, a child could use this. And that is the point. It took 5 years to get it to be that simple, and we worked very closely with engineers and others at the University of Wisconsin to get it to that point. It started being designed in 2015, and we worked with the public to do iterative redesigns in order to make it simpler and simpler. It allows for customized mapping, so if you can use Google Maps, you can use this tool. It's free and open to all. We have over 1,000 downloads every week of the full data, which include 39 million data points. Most of them are outside of research audiences. These are to public health, hospital, and industry groups. And it has over 10,000 hits a week in the mapping app. It's been extensively used in research, industry, and community, and policy. So this is what it looks like, very simple. You can pick your state. You can immediately see in a heat map what areas are more disadvantaged, what are least, and you can zoom in to find your way. So again, it's supposed to approximate what you see on your smartphones. Lots of groups are using it. We have a number of industry partnerships that have been established with the University of Wisconsin, including with EPIC. They're very interested and have already started linking the ADI. We have worked with Blue Cross Blue Shield plans across the country. They change the way they think about responding to quality on the basis of these metrics. Additionally, the Centers for Medicare and Medicaid Services is using this metric as its primary health equity benchmark to change the way payments are flowing to these sites. So if you are caring for a patient that is coming from one of those high ADI areas, you get more money to do so. This just started in January of 2024, and it is revolutionary for the United States. Never before have healthcare payments been adjusted by an equity measure. We've worked with Florida Medicaid and a number of other Medicaid programs, including the state of Maryland and Puerto Rico. We also were interested in use case, because in data democratization, we put very few strings on this tool. And so we sent out a survey just to users that had registered and asked them, how are you using these data? We were able to map many of the areas. In Puerto Rico, for instance, it's being used for mosquito mitigation, to target resources for outreach, for where do we send the staff. Some selected use cases here, number of children's hospitals across the United States deciding who needs additional resources for child safety seats and booster seats and helmets, or different types of groups that are thinking about scarce resources, like during the post-surgical setting, and deciding which patients to receive additional visits. There even are projects out of Chicago that are deciding where to send exterminators most effectively in order to decrease rat populations within certain areas of the city. So a wide array of different activities. The NFL program, the National Football League, is also using the metric to decide for its foundation, the NFL Play 360 Foundation, which areas are most eligible for playground grants, for putting playgrounds within different neighborhoods. So very exciting. This is just one example. I could give you hundreds. This was from a press release just in May of 2023, in which the Milwaukee Water Works has a new equity prioritization plan to try to be more efficient about their lead pipe removal and mitigation. And they are using the ADI to do this in combination with childhood testing of lead levels. But within Milwaukee, reaching all of the children that need testing is incredibly difficult. The infrastructure has not been able to do that. So they are going now street by street, which is much more efficient than going one house here and one house here and one house here. So it's allowing them to move forward. This has been well received by the populace of the city, and they are now using this philosophy in other areas as well. So ultimately, I would say we all do research to change the world. That's what we're here for. We are here to do research that is actionable and leads to change. And putting tools in the hands not only of researchers, but of community members, of health systems, of industry groups, and public health departments really helps these stakeholders to take action on their own, to amplify their own needs, and to support the work that needs to move forward. So data democratization is very important. It is expensive to do, though. It is not easy to make data open to everyone, but it is well worthwhile. So, and of course, this is what we want to see. So in conclusion, I hope I've convinced you that the social exposome is important, and addressing brain health disparities, as we're here to talk about the theme today, requires inclusion of many different features of the exposome, but also the social features. The neighborhood study provides really exciting new research infrastructure, and we are very, very hopeful and excited to move into the more open data phase of that, to be able to safely and securely share data, to build different partnerships with many of you, hopefully, and with others, to open pathways towards new mechanistic discovery. This is a disease without a cure right now. We have treatments, but we need these new areas of discovery to move forward. And then finally, democratizing social exposome data leads to real-world change. So I encourage you to think about that as you move forward and to think outside the research bubble. So these are our sites for the neighborhood study. Many of you are at some of these sites. So this is the largest study I've ever led. It includes 78 key personnel. Again, I have to thank the National Institutes on Aging for funding this, and of course our participants and the many brain donors who have truly given a massive gift to science, and all sorts of funding. So thank you very much.